there's students that are out for like medical things, so rather than trying to make up things later, I'm going to be recording on here, so that's all.
Wait, who's out?
Not in this class.
Oh, we can say hi.
Virginia. Virginia Hagen.
Hello, Virginia. Hi, Virginia.
Yes. Okay, so cellular respiration. It's okay. You said it, and I'm sure, feels very loved. Cellular respiration is the process of breaking down glucose in order to make what?
ATP.
ATP. So we are going to be breaking down glucose to make ATP, which is our form of usable energy.
Who was the other
Tropical. All right. So we know that in metabolism, we have all of the chemical reactions inside of a living thing, making and breaking things. Most of the time when we talk about metabolism, we're talking about breaking things down. In this process of breaking things down, we're going to be moving energy around. And when chemical bonds change, energy is moved around. We move that energy around and put it into ATP. And so the creation of ATP, we are packaging the energy in that molecule, breaking one bond and moving energy into a different bond. All right, we talked about this like either last week or the week before. Endergonic and exergonic reactions. Talk to somebody near you. Try to remember what those words mean. Work it out with some root words. Something that's
I don't know where that's a
Exit.
action.
Ender is not ready.
You know what we're talking about. Endoplasm.
Yeah.
Oh, it's like oxygen.
Exercise.
All right, so I'm hearing some of the correct root words, some of the right ideas. What's the overall point of this unit? What are we talking about?
ATP.
Okay, photosynthesis, ATP, energy. Okay, so you got the in and out part. Energy is the other part of this. So an endergonic reaction is one where we are putting energy in to this thing. So we have a net input of energy. Energy is going in to our products. We are putting energy into this reaction, packaging it in the products of the reaction. Exergonic is the opposite. So when you were talking about endo and exocytosis, you're talking about things going into cells or out of cells. We're still talking about in and out. It's just a matter of, are we putting energy into things or are we allowing energy to leave things? Okay, so an exergonic reaction is one that releases more energy than is required to get it going. Alright, so in our two major reactions of this unit, cellular respiration and photosynthesis, which one would be endergonic?
Oh, endogonic would be
Photosynthesis.
Photosynthesis is an endergonic reaction. We have this input of energy that we package into our products of glucose. Some oxygen gets made, but that's not really the point of it. Alright, and our exergonic reactions is cellular respiration. We have a net output of energy from our reactants to our products. Okay, so we've talked a lot about the endergonic part of this. How do we package energy into glucose molecules in photosynthesis? Now we'll talk about how do we move that energy into different molecules into ATP. This energy can be moved around either in the presence or absence of oxygen. So cellular respiration can be aerobic or anaerobic. Today we're going to focus on the aerobic processes. Aerobic means that it requires oxygen. So these are reactions that take place when oxygen is present. The other thing that is necessary for these aerobic processes, which is very important, is you also need mitochondria to actually make use of the oxygen. Okay, so this is something that can happen in eukaryotes. Why couldn't that happen in prokaryotic organisms? They have no mitochondria or any other membrane-bound organelles. So aerobic respiration is only going to happen in eukaryotic organisms that have oxygen and that have mitochondria. Other organisms can still break down glucose. Why would they still need to break down glucose?
Because every cell needs glucose.
Every cell needs energy, usable energy. Every cell needs ATP. And so other cells can still use glucose and break it down to make ATP. They just do it a little differently. This is not going to be nearly as efficient. We don't get nearly as much ATP out of these processes. Okay, so today we're going to focus on aerobic respiration. Aerobic respiration involves many steps. Not every single one of those steps is going to use oxygen, but the overall process does. So we call this overall process aerobic respiration. And what we're doing in this process is breaking down glucose. We are breaking carbon-carbon bonds, and we are moving some energy into different molecules. Okay? So this is a multi-step breakdown of the carbon-carbon bonds of glucose in order to release stored energy and package it into new molecules. And to repackage it. By making new molecules. What new molecules are we going to repackage that energy into? That's going to be a temporary thing. Our electron carriers will temporarily move the energy around. Our end goal is what? ATP. So ATP is our end goal. But we are going to use a lot of those electron carriers to get this process accomplished. All right. We still need that. What are you guys doing? What's happening? Okay. Lovely. All right. Our first step, whether or not oxygen is present, whether or not you have mitochondria or not, the first thing that's going to happen. What's going on computers back there? Is it biology related? Okay, let's stop that then. Thank you. The first step of cellular respiration, of getting the energy from glucose into these other molecules, is called glycolysis. This is technically an anaerobic process because it is not using oxygen. In fact, you don't even need mitochondria to do this. This happens in the cytoplasm of the cell. This happens outside of the mitochondria entirely. So things that don't have mitochondria can still do this. What we're going to do here is break our 6 carbon sugar, of glucose into 2 3 carbon molecules. So this is happening in the cytoplasm. You don't need mitochondria. You don't need oxygen. Anybody can do this as long as you've got the right enzymes, which most things on the planet do.
How would something not have enzymes and not have mitochondria?
Because they have other ways of breaking down sugar. So they have to have some process that does this, but it might look a little different. Sydney?
What does the oxygen do that means that it's required?
We will get there. It's the very last step. Oxygen is basically the end home for all of the electrons. It takes all of the electrons that get moved around in this.
So it's needed to...
It's needed in the step that makes the majority of the ATP, but that is our very last step that we'll get there. All right, so in glycolysis, we have sugar splitting. Glyco, meaning sugar, like glucose or glycogen, and lysis, to split. So in this process, we are literally splitting sugar. In this picture, we've got 6 little circles. What do we think these circles represent? Carbon in a glucose molecule. There are 6 carbon atoms in a glucose molecule. In this process, we are going to break that 6 carbon molecule into 2 3 carbon molecules called pyruvic acid, or also known as pyruvate. If you see pyruvate, it's the same thing. In order to get any reaction started, some amount of energy is required. So you have to have some amount of energy input. That's your activation energy. This is what's needed to get the process started. So we're going to invest a little bit of ATP. We're going to invest 2 ATP to start this process to break my glucose molecule in half. But in return, I'm gonna get 4. So I get a net profit of 2 ATP. I gain 2 ATP in this process. I spent 2 to get 4. So overall, I have a net gain of 4 ATP. Excuse me, of 2 ATP. When we're looking at this picture, these things are your reactants. And these things are your products. We need a little bit of ATP to start this process. We need an electron carrier to pick up the super energetic electrons that are released from our glucose when it gets split. And we need some ADP if we're going to make ATP. So we break our glucose into 2 3-carbon molecules called pyruvic acid. In that process, we release some energy that gets packaged into ATP, and we also release some really energetic electrons that get picked up by our electron carrier of NAD+. So if NAD+ is picking up electrons, is it being oxidized or reduced? Reduced. No, you were right the first time. It's being reduced. Remember, oil rig reduction is a gain of electrons. So our glucose gets oxidized in this process. It loses some electrons, but somebody picks them up. Our NAD+ is reduced. It gains electrons from that glucose. It also picks up some hydrogen ions. So it gets these super energetic electrons that the glucose has lost, and it gets some hydrogen ions. That pink is really hard to read on the black. Yeah. Let me, it says glucose, let me just rewrite that. Glucose is oxidized. NAD+ is reduced. Glucose is oxidized. NAD+ is reduced. It gains electrons and hydrogen ions. Those are very energetic. The electrons contain energy. They're bouncing around. They're super energetic. Are the fidgets distracting more than they're helping with
No, they're
focus?
helping. They're helping.
Okay, I'm seeing a lot of passing and side talking about them. Ernst, do you have a question? Or you just, okay. I thought your hand was up. Okay. All right, so we broke our glucose in half. We made these 2 pyruvic acid molecules, but they still have a lot of energy left in them. We release some energy by breaking the bonds in glucose and moving those things around and making some new bonds to make these things. But we still have a lot of pyruvic acid that contains energy. We still have a lot of energy that we have not released from this molecule. So what are we going to do? Release it. We're going to break some more bonds. We're going to make some new things. Okay? So we are going to break some stuff. Break more bonds. Every time we break those carbon-carbon bonds and we remake new bonds, we're moving energy around. We are moving energy from one thing to another and releasing some energy in the formation of those new bonds. So we still have some potential energy that is contained in these molecules. We need to do some more processing in order to get that out. So we're going to break it again and move some energy again. We do what's called pyruvate oxidation. We take those pyruvic acid molecules that were made, but there were 2 of them, so we do it 2 times. Each one of those gets broken a little bit more. We break another carbon-carbon bond. In this process, we make a new 2-carbon molecule called acetyl-CoA.
Yes.
And it still contains energy. That we haven't released yet, but we got a little bit more energy out. We broke off another one of our carbons. This carbon, if my carbon-carbon bonds are where the energy is contained, Does that carbon have any more energy I can access in CO2? Are there any carbon-carbon bonds there? No. The CO2 has given me all I can get. This is now waste. This carbon is not attached to other carbons anymore. So this is waste. And I make some more waste here. So we're done with this. Because it has no more carbon-carbon bonds with all that energy. So we're done with the CO2 here. This is where 2 of the CO2s in my waste, in my reaction, come from. 2 of them are coming from pyruvate oxidation. I break a carbon off of this pyruvic acid. I get CO2 waste. I break one off this one. I get another one. There's 2 of my 6 CO2s that I've made in this process. Did I make ATP in this process? No. So if breaking bonds can allow me to move this energy elsewhere, where did the energy go?
Acetyl.
There's still some contained in acetyl-CoA, but when I broke off that carbon, yeah, my electrons that I allowed to move out of my pyruvic acid, those contain energy. So my NAD plus picks up these super excited, energetic electrons. I'm not making ATP, but I am still releasing some energy stuff, some very useful, energetic things that I can move around to other parts of this process. So in pyruvate oxidation, I did not make any ATP directly. So I didn't make the end goal product, but I did release some of that stored energy, some of the potential energy. That energy, instead of getting packaged in ATP, moved and was put into my electron carrier. So the high energy, it picked up my high energy, picked up high energy electrons and some hydrogen ions that were released when pyruvic acid was broken. Okay, so no ATP got made, but I did still release some energetic stuff. I did still accomplish something here. I just moved some electrons around. My 2 carbon molecule that I still have, my 2 acetyl-CoAs. I broke my 2 pyruvate a little bit, broke some carbons off of them, but I still have these 2 carbon molecules, acetyl-CoA. They still contain some more stored energy, some potential energy. So what am I going to do to those?
Break those 2.
Break it again. I'm going to break the last carbon-carbon bonds that I have. I'm going to release that potential energy and put it into this other molecule. Or these other molecules, I should say. So this whole process that we're talking about is breaking little bits off. Remember, enzymes are controlling the rate of these reactions to allow for the slow and controlled movement of energy between molecules. So it's not just spontaneously releasing all of the stuff. Ian and then Sydney.
So acetyl-CoA contains more energy than pyruvic acid.
It doesn't contain more than pyruvic acid, but it still contains some. So each step of this breakdown, we're releasing little bits. Glucose contained the most potential energy. Then we broke it in half. Those things still contain some amount of energy. These acetyl-CoA still contains additional stored energy. I shouldn't say more than the other things. Still contains stored energy. So there is still some in there. It's not more than the other 2 things that we broke already.
Is it less?
It is less, yes, it is less. Each time we're breaking this molecule and making it simpler, it's sequentially releasing some of that stored energy. We're putting it into those other things like the electron carriers and the ATP. Yeah. Sydney?
Where's the phosphates in this?
Realistically, if you are making ATP, it, like if we go back to the glycolysis part, it just said ADP is going in. Phosphate would be needed too if you're making ATP.
Where's the phosphate come from though?
From previously used ATPs. When we use the energy from ATP, it breaks off a phosphate group. We have ADP and phosphate. Those can go back to the mitochondria or to the cytoplasm where glycolysis and then the other parts of cellular respiration happen to get put back together. So it's this continual recycling of molecules. All right. Now we do our last carbon-carbon bond break in the citric acid cycle. This is also sometimes called the Krebs cycle. Named for the person who studied it. Citric acid cycle is more descriptive because it does contain a molecule called citric acid. But if you see Krebs cycle, it's the same thing. So we still have that last 2 carbon molecule of acetyl-CoA. We're going to break that last carbon-carbon bond. And in this process, we have broken our last carbon-carbon bond. Now we have some CO2. We've separated those 2 carbon atoms. Those are now waste. We're done with these now. We're done with them. They have no more carbon-carbon bonds. So we have gotten everything we can out of them, basically. So now those are waste. So there's 2 more CO2s from here. If I have 6 total, I got 2 CO2s from pyruvate oxidation. How am I getting 4 from this?
There was 2 of them.
Because there were 2 of them. There were 2 acetyl-CoAs. So this happens with both of the acetyl-CoAs that we made. This, just like pyruvate oxidation happened to both of those pyruvic acids, this happens to both of the acetyl-CoAs that are made. All right, so we make this waste product, CO2, we're done with it. It's out of carbon-carbon bonds to release any energy. But we made some good stuff here. We made some stuff that we are going to use in some capacity. ATP is good because ATP is how we can use energy in the cell. So this can go be used to do work in the cell. We've also made some energetic stuff. We have contained some super electrically charged, some very excited electrons and some hydrogen ions in this and this. And so these contain stored energy also. They have super excited, very energetic electrons. That they are carrying. Burns, did you have a question?
No, I was saying, so the ATP is just used to like energize stuff?
Yeah, it's now, that's one of our like directly useful products. We make ATP, it can go be used by a protein to do something. Yeah.
Okay, so 2 questions.
Okay.
First of all, if we're not using ATP during any of these processes, what is using ATP? Like, what exactly is using it?
So all of the other things in your cell, basically. And we did use a little bit of ATP at the beginning of this process to start glycolysis. So some of it is used in this process. But when you're looking at like moving things around the cell or active transport or any of the other cellular processes, moving the cell around, all of those things require energy. So anything that a protein is doing in your cell to make everything run, that's where the ATP is going.
Got it. And then secondly, if there are 6 carbons in the beginning and 6 leaving, then how is it recycled? Like, how is there always 6 if there's not always 6?
There is always 6. So we put 6 in with glucose. We let out 6 with CO2. Those are waste products. That's done. But that was 6 in, 6 out. We have accounted for all of the carbons. So we have not like created or destroyed carbons in this. We just changed what molecule they're part of. They could be recycled by a plant, and we can make more glucose in photosynthesis. But CO2, we had carbons that were in one molecule in glucose. We broke it apart. Those 6 carbons are no longer bonded to each other. They're in this different molecule. But it's the same atoms. They've just been put into different molecules.
Okay, so why does it keep needing carbon if there's why does it keep needing carbon dioxide?
We don't need carbon dioxide.
The plants.
Plants need it so that they can make new glucose. They use the glucose and do cellular respiration also. So they break down some of the glucose that they make. And if they want to make more energy, they take that solar energy and put together new CO2 molecules into a new glucose. So it is a continual recycling. It's just a matter of the energy moving around. When we make glucose in photosynthesis, or when plants make glucose in photosynthesis, they're taking CO2s and the carbons that are not bonded together, and they make this molecule of glucose where they are bonded together. Now it contains energy. And move the energy from the solar energy into this chemical energy. Then we can do cellular respiration and move that energy into another form of chemical energy, into ATP. So it's just this constant moving of energy. And carbons are kind of the vessel to do that through. Yes?
How is planting trees good then? If like the carbon that comes in goes right back out.
Because we are releasing lots of CO2 in the form of fossil fuel combustion. And so if we want to take in some of that CO2 and get rid of some of the greenhouse gases, we would need more plants to take in more CO2.
Don't they just put it right back out?
To some extent, but they also grow. So some of the glucose that they make is put into their cells, into their tissues. That's how they get bigger. It's like cellulose, which is what makes up plant cell walls. That's glucose. It's just been bonded and chained together. Yeah.
So like they just take in CO2 and only let out some of what they can.
Right. They let out some of it through respiration, but a lot of it stays part of the plant. Yeah.
How do plants die?
Same way anything does.
Which is,
Old age, sickness, damage.
How do plants die of old age?
yeah, like what is the sick, like what,
Essentially, all aging is
In your cells?
DNA degradation. Molecules
what,
start to
meaning what?
degradation. It starts to kind of fall apart. For now, we're going to move on from old age and dying, though, because that's a very complicated process. Ultimately, the ends of your DNA start to get messed up, and you get worse at making new things. All right, so here's what we've accomplished so far. We have broken our glucose into progressively simpler molecules and released energy along the way. That energy has gotten picked up and put into different molecules. So this and this, those are directly useful. These can go get used in the cell. Same thing here. They'll get used by proteins in the cell. But I also made a bunch of other stuff. I made a bunch of other energy-carrying things. Those need a purpose. We need to do something with all of these. So all of these contain energetic electrons, and we need to do something with them. That needs to go somewhere. We need to do something with this energy. And with the electrons. So we've just been loading and loading and loading these electron carriers up with stuff. They have to take it somewhere. If they're an electron shuttle, they have to drop it off somewhere. Otherwise, we just loaded up shuttles and sent them off into the abyss with nowhere to go. And eventually, you would run out of shuttles. You wouldn't have anything to pick up new electrons if you wanted to break another glucose. So they need a destination. They need somewhere to drop off their cargo. Where they're going to drop off the cargo. So far, we've made 4 ATP. That's not bad. We used 2 to break this. We made 4 out of that in total. So we had
I thought it says 4 from that.
those are carbon dioxides.
Oh.
Yeah. So in glycolysis, we spent 2 to make 4 ATP. So in total we made 2 there. We make another 2 in the citric acid cycle. So in total we've made 4 ATP. We have now made 6 CO2 at this point. We've broken all of the carbon-carbon bonds from the glucose that we started with. Okay, so 4 ATP is not terrible, but we can do better. Do those 4 ATP contain all of the energy that was released from all of those bonds that we just changed? No. Where is the rest of it? Yeah, the shuttles. No. We have a ton of energetic stuff, so tons of energy is being carried in the electrons and hydrogen ions in the shuttles that we loaded up. So we have all this unused stuff that we have moved into our shuttles that needs to do something. We need to do something with it. We've made a ton of these shuttles. The exact number is not super important, but it's more than 4 ATP. We made a lot of these things. We loaded them up.
Yeah, I was gonna say, isn't like the range 24?
Yeah, there's a range based on enzyme efficiency and environmental conditions.
Wait, so
Oh, we watched that last night.
Yes.
does photosynthesis and cellular respiration make ATP, or just
Photosynthesis makes ATP in the light-dependent reaction, but then it uses it immediately. So photosynthesis does not make ATP at the end result. It makes glucose as our end result.
okay?
Cellular respiration makes ATP as our end result.
So the ATP is just bonds being broken.
The ATP is a nucleic acid that has stored energy in it. It's just a molecule that can be used to move energy around.
Okay, so if it's being stored and you have to use it to bond more atoms together, then I still don't get where it's coming from. If it's making the same amount of reactions as it started off with, how is energy coming from it?
We're not making energy, we're moving energy. Energy can't be created or destroyed, we're just moving it around. So in photosynthesis, we moved energy from solar energy into chemical energy in glucose. In cellular respiration, we're moving energy from glucose into ATP.
Okay.
We're not creating energy, we're just moving it around. Okay. So, what are we going to do with all of this energy? We are going to use it to power the enzyme ATP synthase.
We learned about that last night.
I know, it's like I give you homework for a reason.
I don't remember what it is.
Yes.
It was 7 days.
ACE for enzymes. Yes. And ATP synthase, enzymes are named for what they do. So ATP synthase is an enzyme that synthesizes ATP.
Sugar ACE.
Yes. Oat sugar ACE enzyme. All right. Are you going or no?
There's 7 minutes left already.
This is true. Okay. All right. So the process of the electron transport chain is kind of complicated. What I want you to get from this, I do not expect you to understand the entire process and exactly how it works. What I want you to get from this is essentially this. The energy that the carriers are taking to the last step is going to power the enzyme ATP synthase, which makes ATP. Okay, so the electrons power the enzyme. The last part of our reaction, we have not used oxygen yet. We have not made water yet. The other part of this, we use the energy to power ATP synthase, and at the end of this process, oxygen takes the hydrogen ions and the electrons that have spent all of the energy. No more energy left. If you add oxygen and hydrogen together, what do you get?
H2O, water.
Water.
That's where the oxygen comes in.
Exactly. Oxygen is where the electrons end up at the very end of all of this. So the electrons get carried to this series of proteins, the electron transport chain.
So now we're talking about aerobic now.
This whole thing, we haven't used oxygen yet, but if we want to move from glycolysis to the rest of these things, oxygen has to be there. Okay.
Oh, I've got a question.
Okay.
Why are they using ATP to make ATP?
They are using a little bit of ATP at the very beginning to get the process going, but once we're after that step, we're not really using ATP. We're using other kinds of energy moving around.
But it says the ATP synthase.
This is a protein. ATP synthase is the protein, the enzyme that makes ATP. So ATP synthase is a protein. It is not ATP. It makes ATP.
Yes, but it's using ATP to make it.
It uses ADP and phosphate and puts them together to make ATP. It's not using ATP. It's using the electrons to to power this pump.
EPC in action.
Oh, so it's like carriers.
Yes, this is where the carriers are going to deliver all of their energy. I'm going to show you a video.
A video.
A video.
Very.
Can we watch it twice?
Probably, actually. It's very short, and I'm going to slow it down.
Isn't like this the video that was on?
It may have been. It's similar. It might not be the exact one. But, okay, so, what we have in this picture. We have these purple proteins. These things are called the electron transport chain. Oops, electron. The electron transport chain. We're almost done, guys. We're almost done.
Walk in. Oh my god.
These are protein pumps that use electrons, energy. Guys, you're going to get real confused if you don't listen. Okay. This is happening in the mitochondrial. This is a membrane inside the mitochondria. Yeah, this is the inner membrane. Okay, so we have these proteins that are going to pass electrons between them. They are protein pumps. They're going to pump things across the membrane. This is ATP synthase.
And it like makes it like turns like a generator and makes
Yes. Yes. Okay, so we have an electron carrier that is delivering energetic electrons and some hydrogen ions to that first protein pump. That protein pumps hydrogens across the membrane. Those electrons still have some energy, so they get passed to the next protein. We pick up some more electrons that are still energetic. They get passed to the next protein. That pump pumps more hydrogens across the membrane. And those electrons still have a little bit of energy, so they get passed again. We have these little relay molecules that carry them from protein to protein. And then this one uses the last bits of energy to pump some more hydrogens across the membrane.
That's only 10 though.
I know.
Why isn't there 12?
The exact numbers are not super important. Don't worry about exact numbers for this. These electrons now need a home. They need somewhere to go. If more electron carriers came, say I broke down another glucose molecule, we would have that same passing. What would happen if nobody was there to pick up these electrons and more electron carriers came?
They would like
It'd
be
just be
jammed.
built up.
You get a traffic jam. You get electrons that build up and then you can't pass anymore because they're all full. So we need somebody to come and take the electrons away.
Oxygen.
This is where oxygen comes. Takes the electrons away with some hydrogen ions,
Wow.
and we made water. Now we can do the process again. We can get more electron carriers from our process to do it again. We can come back and get new electron carriers to do the same thing. Drop off electrons, pump hydrogen ions. But what we've done is we have built up a very high concentration of hydrogen here and a very low concentration here. We pumped all the hydrogen there. Hydrogen does not want to stay here. It wants to go from high to low concentration. Hang on. The only way to do this is through this guy, ATP synthase. The hydrogen goes through, it spins like a generator, and puts together ADP and phosphate to make ATP. This makes 32 ATP. That is a lot more than 4. This is where all of the money gets made.
Makes sense.
Why do plants need water then?
They need water to make CO to make glucose. All right, this is a summary.
Isn't it
This is
making
your overall
water?
process. No, they split water to make oxygen.
But it's just an
This makes water.
yeah.
They are reverse processes. Photosynthesis and cellular respiration are reverse processes.
No, I'm saying, okay, let's say it starts off.
Wait, that makes sense. That kind of.
I don't know why plants need more water if there's always water.
Okay, so
That makes
it's the same idea.
I don't understand the cycle. But I understand that. I understand just like a different part. But what was that? What was that she just showed? What was it? That was the ATP synthase and the cyclic cycle.
Cycle
Cyclones.
Cyclochromes.
Is that cyclones?
How you doing, bud?
Oh, Ian. What do you have? Would you mind waiting? I have freaking. Oh, I have history. Constance, do you have history?
Yes, it's boring. You do your scrolling credit.